Under physical requirements, we're going to cover temperature, pH, the physical effects of water, and associations and biofilms. First, let us talk about temperature. Now, remember that enzymes operate at optimum temperatures. Those are temperatures in which the enzyme, the molecules of the enzyme, are moving at the right speed to work at the fastest temperature. Also, we have protein structure. There are structural proteins that aren't enzymes, that also have optimum temperatures. We also have lipid fluidity. When you get below a certain temperature, they kind of set up like butter, which isn't really good for a cell membrane. If you get too high of temperature, then the lipids are moving too fast and they break up. So all of these enzymes operating at an optimum temperature, the stability of the protein structure, and lipid fluidity lead to optimum growth temperatures for different microbes. With the maximum temperature, that means that if you go any higher than that, the organism is going to die. The enzymes denature, the proteins denature, lipids start solubilizing. Minimum temperature, that's the minimum temperature at which we stop having growth. So these are the brackets, so to speak. We've got our high range, we've got our low range, then we have our optimum. And our optimum, within the maximum and the minimum, is where we get the highest level of growth. In other words, where is the bacterium happiest? Where are they most comfortable, if we can apply human concepts to bacteria? Within this, we have come up with terms for different organisms based on their optimum temperatures and their temperature ranges. Let's start with these psychrophiles. These are organisms that are happiest, that grow best at lower temperatures. So we can find them growing in glaciers and ice and deep sea temperatures, but for most species of psychrophiles, their optimum temperature is about 10 degrees C. You get much above 20 degrees or a cool room temperature and the proteins start denaturing. Mesophiles are those organisms that are comfortable in the same range that humans are. So we go from about 15 degrees C, which most humans aren't comfortable. You're not going to run around in your bathing suit outside when it's, that cold. But then it gets much above 45 and you're sweating and you're dying and you're not very happy. So mesophilic microbes are the same way. They can survive at about 15, they're not happy. They can survive up to 45, they're starting to die. They're most comfortable between about 30 degrees and 40 degrees. Thermophiles are those organisms that like temperatures that are above that. Their optimum range, now realize this is an average of the species, they're comfortable from about 60 to 70 degrees C. That's getting really hot. Then there are the hyperthermophiles, those that like temperatures that even make the thermophiles uncomfortable. Pathogens are the most commonly cultured examples of mesophiles. They like the same temperature that we do. They grow best at body temperature. Makes sense. Thermoduric. These are organisms that can survive brief periods of higher heat. Well, why would we give a special name to that? Well, it's because in food preparation, you have to watch out for the thermoduric organisms. They can survive improper canning, pasteurization, and they lead to food spoilage. Now, one of these that is thermoduric is the organism that causes botulism. You gotta watch out for those nasty Clostridium botulinum endospores. Now to talk about pH. Remember that enzymes and other proteins are sensitive to changes in pH. This is because their tertiary structure and their quaternary structure can be maintained by ionic bonds between side chains. Well, if you change the pH, then you break those ionic bonds that are holding together the structure, and the enzymes and other proteins denature. So we say that a particular species of organism has a range of acidity, which is the preference and tolerance that that particular organism has for that pH. Now, neutrophiles are those organisms that can tolerate between about 6.5 and 7.5. That's pretty neutral. Now, we could talk about alkalophiles. Up until recently, we didn't know of very many species of organisms that could put up with high pH. But they hit the news a couple of years ago when a researcher was finding organisms that were growing in alkaline lake beds that appeared to be using arsenic in their DNA instead of phosphate. Well, that's now called into question. But those are alkalophiles. Those organisms that prefer a pH that is above about 8. Acidophiles are far more common. These are organisms that prefer a pH at about 6 or lower. Now you have obligate acidophiles, and those are organisms that die if the pH approaches 7. So you have to keep them in a low pH. Then there are tolerant acidophiles. In other words, they can survive a low pH, but they really prefer this neutral pH. So they are tolerant to the low pHs, grow faster if they're in a neutral pH. Sometimes we can combine these categories. If we take a look here, here's an overhead view of Grand Prismatic Spring in Yellowstone National Park. It's a beautiful area. If you ever get a chance to go see it, make sure you stay on the boardwalk. And here's an eye level view. It's very hot and it's very low pH because the water is coming through volcanic areas where a lot of hydrogens are added to the system, and it's very hot. Well, we have hypothermophile obligate acidophiles that grow in these hot springs. Isn't it lovely how we can clump these terms together? Let's talk about osmotic pressure. So remember, when we're talking about osmotic pressure, we are talking about hypertonic, hypotonic, and isotonic solutions. Make sure you review these so that you know what the terms mean. Most organisms require that they grow in 3% salt or sugar solution to be happy, to grow at optimum growth levels. So remember, when we're talking about osmotic pressure, it can be ions or it can be large hydrophilic molecules that can't easily pass semi-permeable membrane. Now, seawater is 3.5, so this is above what most organisms that we are used to culturing can put up with. So we would consider them to be obligate halophiles, anything that requires more than 3% salt to be grown in. So marine microbes and microbes that we pull out of extra salty environments, such as, say, the Great Salt Lake and our neighbor to the south, or from the Dead Sea, or from salt evaporation pools, we would call these obligate halophiles if they are unable to grow in a solution that has less than 3% salt. Now, halo means salt. If they were obligate osmophiles, then they would require 3% or more of either salt or some other molecule that is providing osmotic pressure. There are some organisms that are facultative halophiles. They can tolerate higher osmotic pressure, but they're really happier at the lower solutions. We tend to find these organisms growing slowly but well in, say, salt-cured hams, anything where there's a high quantity of salt or sugar. Now, there is another type of pressure other than osmotic pressure. It is barometric pressure, the pressure of the atmosphere or the water on top of you. So, we're talking about deep sea organisms. When we're talking about living in the deep ocean, if you go down 10 meters, that's the equivalent of another atmosphere, or the weight of air at sea level. So they would be living at 2 atmospheres. If you're at 100 meters, you have another 10 atmospheres. So we call these organisms barophiles, baro for barometric, barophiles, because they depend on pressure to maintain the 3D structure of the macromolecules. So imagine this. You're on a research vessel out at the Marianas Trench, and you send down a collection bottle down to the bottom of the Marianas Trench, which was about 3 miles below the surface. and you pull it up really quick. What's going to happen to all of those poor little sea creatures and microorganisms? That's right, they're going to pop. So when we study these organisms, we keep them under high atmospheric pressure so that we can mimic their natural environment and they don't pop.